Hi everyone, good afternoon guests, and welcome to our gene editing webinar. My name is Declan, and on behalf of What's Global, I'd like to thank you all for joining us today. Today, Eric Paul Bennett, Associate Professor of Copenhagen University and Senior Advisor of NovoNordisk, will be presenting on gene editing on the target's outcomes. Are Indels the only outcomes to be expected? After the presentation, we will have a 5-minute Q&A session. Please submit your questions to the chat option on the bottom of your menu display during this. The full recording of the webinar will be available to download on our secure website, and the details will be sent to you after the webinar. So without any more delays, I'd like to get started with Eric's presentation. So Eric, over to you.
Can everybody hear me now?
Yes, we can hear you, Eric. Brilliant. Over to you.
Perfect. Thanks for the opportunity to present some of our data. And what I would like to discuss with you today is what nature and type of outcomes to be expected after a given genome editing experiment. So shown here are the four most commonly used modalities for genome editing. And as you can see, they are very different in the way the molecular buildup of those molecules. So for instance, zinc fingers and talens are modular nucleases, both fused to a FOC1 nuclease, which is basically the activity that cuts the DNA at a given target site. The meganuclease, a different framework completely, and then of course CRISPR-Cas that works in a different fashion, guided by the guide RNA that we'll come back to in a second. But most importantly, what I'm trying to illustrate here is that the way they cut the DNA at the target site gives rise to different overhangs. In the case of zinc finger and talens, they will generate a 5' overhang, and in the case of the meganuclease, a 3' overhang, and in the case of Cas9, predominant blonde end cut. And this will be seen by the cellular repair apparatus differently. So suggesting, and what I'm trying to point out here is that the outcome relates to what kind of modality you are using for a genome editing experiment, which you should be aware of. Now, the next thing that will impact on the outcome is also the way we deliver these nucleases to cells or tissues or whole organisms for that sake. And there are different ways to deliver. Plasmid-based approaches are commonly used in the form of DNA or delivery by RNA. A more recent way to deliver the nucleases to cells is by protein delivery. And then encapsulation technologies using nanoparticles and the viral delivery formats also are ways of delivering these tools to cells. And they also impact on the dynamics, but also the profile of outcomes to be expected after a given genome editing experiment. And last but not least, when we then deliver those to cells, the way we detect those events will be very important to what we are able to basically detect and see. So the methods available for this, I will come back to, and there are some recent developments that will give rise to a broader view of the outcomes to be expected, which is the major point of my presentation today. So obviously the primary editing outcome after genome editing is a double-stranded break formation at the target site. And if you're targeting the coding reading of a gene, that repair by the cell can be perfect and cause a retain of the reading frame, of course. And then you can have the effect that you will generate insertions or deletions at the target site that could be divided by 3 bases. So basically retaining the reading frame, potentially retaining the function of the protein of interest. And then, which is of main interest to many of us, is when we get out of frame caused deletions or insertions at the target site, when this the number of bases are non-dividable by 3, we disrupt the reading frame of the gene, generating what we commonly know as knockout at the target site. And these are most obviously the indels we would like to analyze and detect when we do a given knockout experiment. These are basically the primary editing outcomes after genome editing. Now, importantly, as I hinted to before, the cellular repair apparatus is basically doing the job in the cell, and this requires a lot of components, repair apparatus components, and the different repair pathways, and the capacity of different cells with regard to the different repair pathways will differ in cells and also impact on the outcome of the editing experiment that you are working with. So this slide just sort of portrays a very interesting survey that was done by Biotechnique recently, where they had a worldwide sort of survey and asked different fields and areas from academia to life science to biotech to ask what the main purpose of giving the CRISPR-Cas application was. And the top 5 answer was gene knockout. Was the highest among the top 5 response they got from these respondents. And this sort of puts the central question when you're doing in particular Cas9, which is the focus of the rest of my presentation, whether a given guide RNA design actually will induce double-stranded breaks equally efficient. So on the top, I'm trying to persuade a given gene, and you have multiple choices of selecting guide RNAs for your experiment. So the question here is it irrelevant which guides you use with regard to whether they will functionally cut the DNA equally efficient? And the next question you may then ask is, how is a given guide actually functionally validated? So how do I know that the guide RNA design I'm actually using will actually cut the DNA at a high efficiency? And I'll point to both of these issues in the slides that I'm presenting in the following. Now, this is just an example we showing a major effort we started a few years ago, where we tried to establish a guide RNA collection targeting 200 human genes that are of our interest. And the strategy we decided to pursue was to generate 4 guides per gene, so that's 800 in total, and then transiently transfect those one by one into cells and then monitor the cutting efficiency at the target sites for each of these 800 guides. And through this endeavor, we selected 200, one for each gene, that experimentally would give rise to more than 30% cutting efficiency at the given target site. Now, what's shown below here will guarantee 16, which is a gene of our interest, is in this case 7 different guide RNA designs. And what I show here is that for 4 of these, we could show that they functionally did not cut the DNA at all. They were functionally inactive, not being able to cut at the target site. And 3 of the 7 had very nice cutting efficiency. And most interesting for those that had a high cutting efficiency, we saw that the +1 base pair insertion was the predominant outcome of the editing experiment. So 30% of the guides that we in this sort of study validated would have no functional activity at all, suggesting that not all guides are created equal, but you need to experimentally validate that a given design actually works at the target site. Now, the +1 feature was a general feature in our study, and this has been corroborated by others in the field also. The basic sort of concept here is that for a functionally very effective guide, the +1 base pair is a predominant outcome. And we saw that in more than 60% of our validated guides, this +1 was actually the predominant outcome after genome editing, which poses some requirements to the detecting methodology that you would put in place, enabling detection of these outcomes. So in this case, the +1 base pair insertion. And we've just in the midst of finalizing manuscript, where we have sort of surveyed the available methodologies for detecting indel outcomes. Going from the top, NGS, which probably could be considered as the golden standard in the field, and moving downwards to Sanger-based methodologies such as TIDE and ICE, to digital PCR, and to an assay that we've developed recently called Indels detecting bioimplicant analysis that I will briefly touch upon, because it seems to be as efficient in detecting Indels as next-generation sequencing, and it has the advantage that it's extremely fast methodology and with a low cost. Another way of looking at how to detect the outcome of, for instance, a knockout experiment is to look at the proteome. So in this case, one could take protein from cells from wild type and knockout experiments, and then do protein extraction and digestion, and then do biomass spec analysis, look at the whole proteome. And in case of a knockout, you would expect that the protein encoded gene that you have targeted, that protein would disappear from the proteome, as exemplified in this particular situation where we've looked at 3 biological replicates from wild type cells and targeted cells, and very nicely you can see that the protein or the gene targeted encoding this particular protein disappears from the proteome. So this is another way of sort of looking at the outcome of gene editing, but not from the genetic level, but from the proteome level. And this is a methodology that's currently being a service from a company called Copenhagen Technologies. Now back to the genetic way to profile indels. Briefly, the IDA assay that we developed is extremely simple in its way of working, in the fact that by using a fluorophore label primer, you enable an amplicon to be fluorophore labeled. Now, if this particular PCR product covers the target site, then after genome editing, you would expect different fragment sizes to be represented in this amplicon population. And by applying that on a DNA sequenator, which basically is a capillary electrophoresis instrument, you, on a single base discrimination level, can discriminate the fragments eluting out of this machine, as shown below here, where we in the wild type peak see a yellow peak in the chromatogram, and relative to the yellow peak to the right, we see all the insertion events, in this case a +1 base pair insertion, and to the left we see deletions going from -1 and down to the lesions that can be detected in the sample. Now this is a quantitative estimation of the individual events observed, in this case in a population of 1 million cells targeted with CRISPR-Cas, and we get quantitative readouts of the individual indel events in that population. Now we wanted to compare the sort of sensitivity of IDA to the gold standard next generation sequencing, and briefly this is all published, but just highlight the fact that we basically can quantify on a given data set that the sensitivity of IDA is similar and in the range of 0.1% as next generation sequencing, which is basically the commonly agreed on detection limit for NGS when it comes to indel detection. We also compared whether the sort of the accuracy of digital PCR with the IDA assay, and recently in a malaria fly experiment together with collaborators from the US, designed an assay where the digital PCR sort of primer probe set covers the target site, where we also have an assay that covers the identical target site. And we could show that the accuracy of both methodologies were very similar in controlled indel spiked samples, showing that IDA actually performs as good as digital PCR in this case for quantifying indel events in this case in heterogeneous populations. Now moving forward to the delivery ways and how that impacts on the profile and dynamics of indel outcomes, we in collaboration with Emmanuel Misakopian from the Sanger Institute some time ago published how the different delivery formats impact on the profile, both the dynamics by which indels occur in the population of cells, but also the profile. And this is all published, but I will just highlight three of these sort of delivery formats: RNP electroporation, transient plasmid transfection, and lentiviral delivery of the same guide in Cas9 to HEC293 cells. And in this panel, we see that the dynamics over time, so 6 hours, 1 day, 2, 3, 4, up to 14 days after plasmid delivery, so transfection to HEC293 cells, about 1 million cells, we see how the indels sort of dynamically emerge in the population of cells. And as you can see, 6 hours after delivery, we cannot really see anything occurring in the population. But 1 to 2 days after, we see the prominent 1+1 base pair insertion appearing, plus some minor indels to the left of the wild type peak. And day 3, sort of the maximum number of indels are observed in the population. And this seems to sort of decrease over time, likely to that some of these cells, they will vanish from the population due to other reasons that I will not have time to go into right now. If you look at the lentiviral delivery format of the same guide in this case, we see that the dynamics by which the indels are generated in the population are similar to when we deliver plasmids. 6 hours, we cannot really see something. Day 1, we see some indels appear in the population, and the signal sort of maxes out day 3, similar to what we observe for the plasmid delivery. And then it's sort of a steady state with regard to indels in the population that remains constant over time. If we then apply selection on the lentiviral transduced cells, we can see that the wild type cells are killed off, and then we see very prominent indel formation in the selected population, as expected. Now, most interestingly, when we deliver by electroporation RNP complex with guide RNA, the dynamics is very much different compared to the 2 other delivery formats. And 6 hours after electroporation, we see a prominent 1 base pair insertion appear. And basically 1 day after transfection, all cells in the population have been targeted or edited at the target site. And there's only a reminiscent level of wild type alleles left in the population, suggesting that nearly all have been biallelically targeted and some are only heterozygous, but the majority have been edited, showing that the delivery format clearly impacts on the way by which indels are formed in cells. Now, this is another example where we, in collaboration with Morten Frodin at the University of Copenhagen, wanted to do in vivo determination of indels in a whole animal. And in this case, we tested a guide RNA in cell culture, and show that the guide is able to generate indels. And then hereafter, we injected by tailbone injection, plasmid guide and Cas9 into the tailbone of a mouse. And then 3 weeks after post-injection, we harvested the liver from the mice to analyze whether indels could be detected in areas of the liver biopsies. And we can see here that in the normal area representing normal liver tissue, no indels could be detected, whereas in the dysplastic tissue, we could see indels appearing. Very nicely showing that this assay could be used for sort of dissecting also on in vivo targeted organs in this case. Now, a commonality of all these detection methods that are used currently for indel profiling is that they are all based on PCR. So in one way or the other, they include this amplification step, which obviously puts some limitations to what you can detect, because these assays will all be limited, that we are looking at only a 150 to 400 base pair window. And this is a little bit in contrast, or the next thing I will change to is to sort of portray to you the alternate outcomes that have not been so much in discussion today yet. And this is a little bit in conflict with previous studies showing that the effects, the outcomes after gene editing seems to be very specific, where only a very limited amount of off-target effects occurring at off-target loci. These are just a few examples demonstrating the specificity of genome editing. But when we look at the various methods, so this is also a very commonly used indel off-target assay, the guide-seq assay. Clearly, it demonstrates that if we look at the experimentally occurring off-targets, and this is not looking at the bioinformatics, This very nice study, essential study, showed that at the given on target, shown in green for MX1, a number of off targets could be shown in cells using this particular assay, and these off targets could not be predicted bioinformatically, as shown in this figure from the paper. In yellow, the bioinformatically predicted off targets, and then the experimentally validated. Some could be experimentally also shown to occur, but there was, in this case, 5 for this particular off target, 5 off targets that could not be bioinformatically shown. So this is one way of using an off-target assay to look at, in this case, minor indel off-targets. But what's becoming very evident now is that other things occur also at the on-target that go beyond these small and minor indels. And one of these is larger deletions across the on-target. And in this particular study, they showed that 20% had deletions from 250 base pairs and extending up to 6 kilobases around the on-target. This is what we can sort of consider as on-target outcomes, unintended on-target outcomes that would not be picked up necessarily by a PCR approach because we're limited to these 100 to 400 bases. And one of the conclusions of this study was that DNA breaks induced by single guide RNAs frequently result into deletions extending over many kilobases. Again, pointing to the fact that you need to have appropriate assays in place to be able to detect these adverse effects. Another study, very recent study from this year actually, also points to the same fact that when we do genome editing, we get these unintended on-target outcomes at the on-target, that is. Most commonly encompassing larger deletions occurring at the on-target, again, cannot be picked up by conventional PCR assays. In this case, as shown here, deletions or insertions up to 2.8 kb were identified. Again, this in a study from mice zygotes, also demonstrating that large deletions, in many cases, by PCR approaches, are not detected, and by using what we call long-range PCR, some of these can be revealed, as shown in this case in the blue bars. But some of the even larger deletions cannot be easily picked up by PCR, not even long-range PCR, and go undetected through a study. And this is also another data set from the same study showing that for this particular target in the mouse zygote also, they observed by whole genome sequencing larger deletions across the target site in many of the outcomes, F1 outcomes of this study. Again, the bottom line here, the frequent large deletions detected are detected in mice zygote injections, and they can extend up to thousands of bases, as seen in the figure on top. So the concept now appears that when we do a given experiment, yes, on the on-target, we can detect the minor indels by the conventional assays that I showed previously. But also what occurs is obviously the unintended targets can also be hunted down with assays such as GuideSeq and others to detect where these minor indels may occur. But on top of that, we have to realize that unintended on-target outcomes are also a commonly seen theme or outcome after genome editing. And these unintended outcomes include large deletions beyond 1 kb up to 10 kb and larger. And also, of course, obviously translocations where double-stranded breaks are induced at the on-target, potentially also at off-targets, giving rise to possibility of translocations. And a lot of recent reports showing that translocations are also a common observation or outcome after genome editing. And obviously the last but not least are the structural rearrangements where due to off-target cutting, larger structural rearrangements may occur in cells after genome editing also. So I think we have to sort of broaden out the toolbox by which we analyze outcomes after genome editing. And one of the novel approaches to undertake for hunting down these unintended, both on-target but also off-target events is could be the solution to use single molecule sequencing for detection of these unintended on and off targets. And the advantage with these technologies are that as opposed to Illumina sequencing, was basically a short read sequencing, less than 500 base pairs you can sequence. Long read sequencing are single molecule, and they can read sequence lengths of 20 kb up to even 200,000 bases have been reported to being able to be sequenced by these platforms. The two platforms that are enable this are Nanopore and PacBio. And they are very different in the way they work. The Nanopore technology is a single-stranded DNA sequencing engine, basically, where sort of a helicase pulls down the single-strand DNA through a pore that is applied on a graphene chip, and it can detect the current changes depending on the bases that are being pulled through the pore, and this can be detected as a readout and thereby sequence. And the other technology, PacBio, is based on basically a polymerase that reads that incorporates fluorescently labeled nucleotides that can be detected and give rise to emission signals that can be detected and thereby sequenced as a single molecule. And I strongly believe that these methods need to be put in place in order for us to get a better overview of the complete landscape of outcomes, not only covering small minor indels, but also these larger structural variations that also may and can occur after genome editing. And both these platforms described here, Nanopore and PacBio, they enable sequencing of single DNA molecules and thereby can reveal whether or not these events occur after genome editing. So coming to an end, I just want to wrap up with some of these events that one should take care of and also investigate. Some of the unintended on-target outcomes will include indels, both the minor but also larger, translocations, larger deletions, and insertions in particular. And this goes also for the off-target, where the same events may occur and for now are not really being disclosed due to the fact that the methods being used for these off-targets will not reveal the nature of those outcomes. And with this, I think my time is up, and I just want to thank the Foundations for Financial Support to our study, Henrik Clausen, the director of the center, and Hans Vandal, the vice director of the center, for support, and Morten Fordin and Emil Mesakopian and Esken Met from the Herlev Hospital for support to the study, and Melbourne Sigma for some of the reagents used in this study. And I thank you for your attention and are open to questions from you.
Yeah, thank you very much, Eric, for this presentation. And reiterate what he said, please submit your questions to the chat option on the bottom of your menu display, and we'll go through them as we get them. So thank you. So there's one here, Eric. So unexpected gene editing outcomes are to be expected at both on and off targets. What are the major issues related to the induction of these issues?
Yeah, it's a very interesting question. I think one of the issues that we are not so aware of is that when we choose a given genome editing tool, we most often select the one that is most potent in cutting at the target site. So the most efficient one is the one we move forward with. Now, needless to say, if it's very efficient at the on-target, it may also be very efficient at the off-target. So the better the cutting at the on-target, the more off-target you could envision you would also create. And as displayed here, if you have these double-stranded breaks occurring at off-targets, you are very prone to the repair apparatus' ability to generate these translocations and inversions and larger structural rearrangements. So I think one of the problems is the way we basically select a perfect genome editing tool may not be the one that it cuts the most efficiently that we should move forward with, but I think more studies need to be conducted to sort of firmly establish this particular statement.
Okay, yeah, brilliant, thank you. So got another question here. What do you think is the best approach to detect large unintended outcomes, whole genome sequencing on long-read platforms? So.
That's a very interesting question. I think it perhaps boils down to a few things. One of all, the first thing is probably cost. So if you can afford a whole genome sequencing and have the resources to do that, this might be a good approach. My take on it is to use different approaches also for these larger effects. So whole genome sequencing could be one, but there's a series of recent publications looking at how we can tackle this problem with methods other than whole genome sequencing. So nanopore and PacBio are two approaches, methodologies that could be used, but there are other ways to tackle these for now unhunted events also. But so basically, I think whole genome sequence is a resource requiring methodology to use, and the question also if the sensitivity is sufficient enough to, in populations or pools of cells, if the sensitivity is high enough to be able to detect these effects.
Okay, brilliant, thank you. So we have another one here. Are there any methods to reduce rearrangements but maintain repair in CIS at the target site?
Sorry, I missed the first part of the question.
Sorry. So are there any methods to reduce rearrangements but maintain the repair in CIS at the targeted site?
Are there any methods? That's a difficult one. I don't really have a full answer for that. I will have to elaborate on that question. I cannot really address and answer that question actually. Not that come to mind really.
Okay,
so
that's
sorry.
That's no problem at all. So we have another one here. So for your +1 BP inertians, is one type of nucleotide, ACGT, inserted more frequently?
Yeah, that's very interesting. Many recent studies have shown that this is the base that is inserted. It's not random. It's dependent on the sequence at the target site. So it will be determined by the cut site, what base will be inserted with the highest probability. So this is definitely not random, but a feature of the cutting site, the targeted site basically, when we talk about CRISPR-Cas, yes.
Okay, brilliant, thank you. Got another one here. So they say, it seems the unexpected on-target outcomes are often asymmetric. I've been considering capture-based technologies targeted on either side to be able to reliably capture these outcomes. Would that work?
Was it a PacBio specific question or?
I don't actually know. It's by Dustin Rubinstein. So I can repeat the question, or?
Yeah. Just repeat. I think I got it, but I just want to be clear on my answer.
No problem. So it seems the unexpected on-target outcomes are often asymmetric. I've been considering capture-based technologies targeted on either side to be able to reliably capture these outcomes. Would that work?
Yeah, that's a very interesting question, and it is true. It seems that the literature suggests that they are asymmetric. So how do you cover both sides of the targeted locus? Probably you would need to implement a strategy where you look from both sides, basically. So there are assays like LAMP-seq, where you basically, it's sort of a primer extension methodology where you sort of extend a primer from one or the other side across the target site to see what's on the other side. So that could be one way to address the issue, to sort of address the cut site from both angles and see what's on the other side. Thereby you could at least identify translocations.
Okay, brilliant. Hope that answers the question. So we have another one here. If a donor template is inserted at the target site, can on-target deletions also occur?
If a donor is integrated, well, it probably depends on the size of the donor. So if you insert a large sort of a gene cassette there, called on-target effects. Well, one of the effects that has been recently published is that for larger donors, it seems to, and in particular if you do a homology-directed repair integration of a donor, it seems that the concatemerization of the donor at the target site is a feature that you see at the on-target. So there you would have multiple copies going into that site, and they could be missed by junction PCRs, because then if you do a junction PCR for the 5' or 3', you would say, we would think if that was positive, that you would have one copy in there. But in fact, you could have, and even more interesting, it seems that they are sort of in head-to-tail insertion events at the target site. So positive junction PCR could just indicate that you had at least one copy, but it would not rule out the fact that you could have multiple copies sitting at the target site. So that could be an on-target effect as a result of putting something in, so a knock-in experiment that you should be aware of. And there again, Nanopore and PacBio would be able to pick up how many copies you in fact had at that target site. So my take on that would be to basically do the junction PCR and then do Nano or PacBio to confirm that you only had one copy sitting at that target site.
Okay, yeah, brilliant. Thank you for that. So, what would in your mind be required for reliable mapping of unexpected gene editing outcomes?
Yeah, I think my first answer would be here that no one assay can stand alone. So I think it should be a complementary battery of different assays that tackle the problem from different angles. Now again, depends on what the aim of the study is. If it's an academic situation, it might not be so big an issue, but if you go to therapeutics or for other things that go into whole organisms, you probably want to do a reasonably thorough study that proves or disproves that this is actually a problem or not. So I would implement several assays, and Nano and PacBio would be one of those assays I would implement for looking at that. But then other like LAMP-PCR that I hinted to before, or other assays could also tackle the same issue from different point of view, different experimental point of view.
Okay, brilliant. So I think this should be the final question, unless anyone sends any more through. What is the latest registration count?
What is the latest what?
What is the latest registration count?
Registration count regarding, I'm not sure what it relates to.
To do the mapping of unexpected gene editing outcomes, I think so. Don't know. Always.
If it's regarding percentage of how many off-target effects occur in a given experiment, if that's the question, registration of off-target effects or what?
I don't have any more information from the question, so you can always go with that if you think it's useful for the audience.
Yeah, because I'm not sure how to address that question. I'm not sure what the question is about.
Okay, that's cool. We can always wait for more information. We have another question through anyway, so we can always go to that one, come back to that one.
Okay.
Would it be reasonable and economical to implement nanopure and or PacBio technology to analyze potential mutant alleles in founder animals?
Now, that's an interesting question. So I know that so the two technologies are very different, as I try to portray, and the instrument requirements are also very different. So the PacBio instrument is, as you could see in the image, man tall high. It's a big instrument, whereas the nanopore is just basically a cigarette holder that carries a chip onto which you apply your sample. So the cost for doing nanopore sequencing is much lower than PacBio. So if cost is an issue, this is a feasible strategy for looking at these larger effects using single molecule sequencing. There's some issues related to the accuracy, but if accuracy is not an issue because you're not interested in the accuracy of the sequence as such, but more what the sequence represents, so a transition or translocation and so forth. Then I think nanopore could be a very attractive sort of single molecule sequencing platform to implement for looking at these unwanted effects.
Okay, brilliant. Thank you for that. So I think any more questions are coming through. So I think I'll wrap this up now. So thank you so much for that presentation again, Eric.
Yeah, and I thank you for your attention.
Yeah,
and of course I will speculate more on this on the conference in November.
Yeah, brilliant. Yeah, so Eric will be speaking at our upcoming 6th Annual Genome Editing Congress taking place on the 4th and 6th of November. So if you'd like further information on the Congress or would like a copy of the brochure program, please contact our marketing department or visit the conference website. On behalf of Vox Global, I'd like to thank everybody for joining us today. And I'd like to say a big thank you again to Eric, who's put a lot of time and effort into making this webinar as interesting and valuable as possible. So I hope you all have a great rest of your day, and we look forward to welcoming you at one of our future events. Thank you.